Imagine wearing a small patch on your skin that continuously checks your health, your glucose level, hydration, stress, or even early signs of infection, without needing a needle or a hospital visit. How is that possible? The answer lies in wearable lab-on-a-chip and microfluidic devices. Tiny laboratories that can fit on your wrist, in a patch, or even inside a piece of clothing. These devices guide microscopic amounts of sweat, saliva, or interstitial fluid through channels thinner than a hair, where chemical sensors measure biomarkers in real time. Why is that important? Because your body constantly changes, and these changes often appear in fluids long before symptoms become visible. For example, a microfluidic sweat patch can detect rising lactate levels during exercise, warning athletes of fatigue or dehydration before it's too late. But how do these mini labs actually work? Inside them, microchannels act like veins, directing body fluids to detection zones coated with special materials that respond to target molecules, such as glucose, cortisol, or electrolytes. The resulting signal can be electrical, optical, or even color-based, then sent wirelessly to your smartphone. The amazing part is how little fluid is needed. Just a few microliters can reveal your physiological state. Some devices even integrate microneedles that gently sample fluid beneath the skin without pain. Example, continuous glucose monitoring patches now use microfluidic sensors to track sugar levels minute by minute, replacing finger prick tests. Others measure pH and sweat rate to prevent heat exhaustion in soldiers and athletes. The next step is even more exciting, combining AI algorithms with microfluidic sensors to predict health issues before they happen. Could your smartwatch one day warn you of an infection a day before you feel sick? That's the goal. By learning your body's unique chemical patterns, AI-enhanced lab-on-a-chip systems could personalize healthcare like never before. These technologies are turning passive health monitoring into active prevention. A future where a few nanoliters of fluid tell the story of your well-being anytime and anywhere.
Explore how wearable microfluidic systems transform continuous health monitoring through miniaturized biosensing platforms. Wearable lab-on-a-chip systems integrate miniaturized sensors and microfluidics directly on the skin, creating a distributed diagnostic network across the body's surface. Let's understand why combining wearables with microfluidics revolutionizes health monitoring. Continuous health monitoring captures trends and circadian patterns that single clinic visits completely miss, providing longitudinal data streams. Traditional clinic visits provide sparse snapshots, while wearable systems deliver dense, continuous measurements that reveal the complete picture between visits. Microfluidic channels precisely manipulate nanoliter to microliter volumes of biofluid using minimal power and reagents through clever physics. Microchannels use capillarity, osmotic gradients, or body motion to drive passive flow through precisely engineered geometries. Integrated sensors transform chemical and physical signals from biofluids into electrical readouts that can be processed and transmitted wirelessly in real time. The sensing pipeline converts molecular information into digital data streams through electrochemical, optical, or capacitive transduction mechanisms. Different biofluids provide distinct molecular windows into health status. Each biofluid offers a distinct molecular signature: sweat for electrolytes, interstitial fluid for glucose and drugs, saliva for hormones, and blood for comprehensive biomarkers. These four primary biofluids serve as complementary information channels, each accessed through specialized sampling interfaces. Channel geometry, wettability, and surface energy are precisely engineered to route fluids without leakage or backflow through Laplace pressure gradients. Channel height directly controls hydraulic resistance, with narrower channels providing higher resistance and better flow control through precise dimensional engineering. Microneedle arrays and porous membranes lower transport barriers, allowing controlled extraction of interstitial fluid from beneath the skin surface. Hollow microneedles penetrate the stratum corneum to draw interstitial fluid into microchannels at controlled rates for continuous glucose or drug monitoring. Materials must match skin's mechanical properties to maintain comfort and sensor accuracy. Soft elastomers like PDMS and silicone gels match the skin's low modulus to prevent motion artifacts and irritation during natural movement. Island bridge architectures place rigid sensor components on flexible interconnects that distribute strain, allowing devices to bend with joints without breaking. Serpentine traces, wrinkled films, and kirigami cut patterns act as mechanical decouplers that absorb strain during motion without degrading electrical performance. Serpentine interconnects uncoiled during stretching, maintaining electrical continuity while accommodating large skin deformations during activities like joint bending. Micro-perforated films and breathable laminates allow vapor exchange to prevent sweat accumulation and skin maceration during extended wear periods. Micropores allow water vapor to escape while blocking liquid sweat, maintaining thermal comfort and preventing skin irritation during multi-hour monitoring sessions. Real-world applications demonstrate the power of integrated wearable microfluidics. A skin-mounted sweat patch with capillary burst valves continuously measures sodium and potassium levels during exercise, alerting athletes before dehydration symptoms appear. The patch collects fresh sweat through porous membranes, routes it through microchannels, and measures electrolyte concentrations with embedded electrochemical sensors. A soft, throat-mounted patch monitors vocal fold vibration frequency and tissue hydration across rehearsals, helping professional singers optimize performance and prevent injury. Accelerometers detect vocal fold vibrations while impedance sensors track local tissue hydration, providing real-time feedback during vocal training and performance. By integrating materials, science, microfluidics, and analytics, wearable systems transform the body's surface into a distributed sensor network for longitudinal health monitoring. Multiple wearable nodes communicate with a central hub that aggregates data streams, applies machine learning, and delivers personalized health insights in real time. Wearable microfluidic lab-on-a-chip systems represent the future of personalized health monitoring. Microfluidic channels handle minute biofluid volumes with passive pumping mechanisms, eliminating bulky equipment and enabling comfortable continuous wear. Elastomeric substrates and strain decoupling architectures ensure devices move naturally with skin while preserving sensor accuracy during daily activities. Continuous monitoring captures circadian rhythms, exercise responses, and gradual trends that single clinic measurements miss, enabling proactive intervention. The convergence of materials, microfluidics, sensing, and wireless communication turns human skin into an intelligent diagnostic interface for precision medicine. Explore the cutting-edge architectures and sensing technologies that power next-generation health monitoring patches. Modern biosensor patches integrate microfluidic channels, sensors, and electronics into a single wearable platform for continuous health monitoring. Let's explore the innovative valveless architectures that control fluid flow without pumps. Capillary stop valves use surface tension and geometry changes to control fluid timing without any mechanical parts. Here's a capillary stop valve. Fluid flows until it reaches a geometry change that temporarily halts flow until enough pressure builds up. Dissolvable films and hydrophobic patches create timed routing, allowing multi-step assays to run automatically. A typical two-stage channel meters a fixed volume into a sensor chamber, then diverts excess fluid to a waste trap for safety. Phase change materials enable thermally triggered flow control without electronics. A wax valve stays solid at room temperature, but melts at body heat after 5 minutes, automatically starting a timed assay. The paraffin valve transitions from solid to liquid at body temperature, triggering flow without user intervention. Paper-based or textile microfluidics use wicking to create low-cost platforms that can run multiple tests over large areas. Multiple sensing technologies work together to capture diverse biomarkers. Electrochemical sensors dominate because they consume little power and can be miniaturized using enzyme catalysis and redox mediators. Enzymes catalyze reactions with target molecules like lactate or glucose, producing measurable electrons that generate a current signal. Field effect transistors, functionalized with AP tamers, can detect proteins and small molecules without requiring labels or tags. Aptamers on the transistor surface bind target proteins, changing the electric field and modulating current flow through the device. Optical sensing leverages absorbance, fluorescence, or photonic resonators coupled to smartphone cameras for colorimetric assays. Combining multiple sensor types dramatically improves accuracy and clinical context. Sensor fusion combines lactate levels with heart rate and temperature to distinguish between physical exertion and infection. Pairing biochemical markers with vital signs provides the clinical context needed to interpret readings accurately. Aptamer-based cortisol sensors are corrected using local sweat rate measurements to estimate equivalent plasma hormone levels. Efficient power and data strategies are critical for long-term wearable operation. Hybrid power systems combine thin-film batteries, NFC harvesting, and thermoelectrics to balance energy needs across different use cases. A battery-free NFC glucose patch remains dormant until a smartphone scans it, then powers up briefly to update the day's glucose profile. Bluetooth, low energy, and intermittent uploads minimize radio frequency heating and reduce privacy exposure from continuous transmission. On-device digital signal processing filters motion artifacts and performs baseline correction before transmitting data wirelessly. Smart algorithms transform raw sensor signals into clinically meaningful data. Models map sensor currents to analyte concentrations via calibration curves with temperature and pH compensation. On-chip calibration standards, phantom fluids, or model-based inference from correlated signals maintain accuracy over time. Time series analytics extract trends, circadian rhythms, and anomaly scores from continuous biosensor data streams. Personalized models adapt to individual baselines, reducing false positives by learning each user's unique physiological patterns. An exercise monitoring patch stores minute-level data locally and uploads only summary statistics after the workout to conserve power. Let's see how these components work together in a complete system. A complete biosensor patch integrates microfluidic channels that route sweat to multiple electrochemical sensors with onboard signal processing. Modular manifold designs allow the same patch platform to simultaneously monitor electrolytes, metabolites, and proteins in parallel channels. Advanced, wearable biosensors represent the convergence of multiple technologies. Valveless microfluidic architectures enable autonomous, multi-step biochemical assays without pumps or user intervention. Combining electrochemical, optical, and mechanical sensors through fusion algorithms dramatically improves diagnostic specificity. Hybrid power sources and edge computation enable continuous monitoring with minimal battery drain and real-time feedback. Machine learning algorithms that adapt to individual baselines provide clinically actionable insights while minimizing false alarms. Explore how wearable biosensors move from lab validation to real-world healthcare applications. Wearable biosensors bridge the gap between laboratory measurements and continuous real-world health monitoring. First, we examine how wearable sensors are calibrated and validated for clinical accuracy. Wearable sensor readouts must correlate to blood or clinical endpoints through cross-fluid conversion using partition coefficients and lag time models. The conversion process transforms sweat measurements into blood equivalent values through mathematical models and calibration factors. Human validation studies compare wearable patch outputs with gold standard venous blood draws under controlled protocols. Validation graphs show how closely patch readings track blood measurements over time during clinical trials. Bland-Altman analysis is the statistical method used to quantify agreement between the two measurement techniques. For glucose monitoring specifically, Clark and Park's error grids evaluate the clinical risk of measurement errors. Interferent testing challenges sensors with urea, ammonia, medications, and variable ionic strength to ensure selectivity. For example, a lactate patch is validated on cyclists with simultaneous blood lactate measurements every 10 minutes, achieving clinically acceptable agreement. An analogy is calibrating the scale with certified weights before using it for commerce, ensuring accuracy through known standards. Now let's explore the diverse clinical applications where wearable biosensors make an impact. In fitness applications, electrolyte and hydration tracking helps personalize fluid intake and prevent heat illness during exercise. Wearable biosensors serve multiple clinical domains, from fitness optimization to chronic disease management and mental health monitoring. In diabetes management, interstitial fluid glucose wearables enable tight glycemic control with real-time alerts and dosing guidance. For renal and heart failure, monitoring weight, edema proxies, and sodium trends supports early intervention before acute episodes. In mental health, cortisol and heart rate variability patterns provide stress phenotyping when interpreted cautiously by clinicians. In acute care settings, hemorrhage risk can be monitored with lactate, hemoglobin proxies, and pulse waveform dynamics. For example, a heart failure patient uses a sodium sweat patch and weight scale, to adjust diuretics under remote clinician supervision. The system works like adaptive cruise control, detecting problems and triggering interventions before critical events occur. Finally, we address the practical challenges of manufacturing, cost, and ethical deployment of these technologies. Scalable fabrication leverages roll-to-roll printing, laser micromachining, and injection molding to achieve low per-unit costs. Roll-to-roll manufacturing continuously produces sensor patches at high volume, similar to printing newspapers or packaging materials. Dry reagent storage with sugar glass or polyolene enables long shelf life without requiring expensive cold chain logistics. Cartridge plus reusable reader designs reduce waste and lower device price points through razor and blade economics. For example, a $5 single-use electrolyte patch pairs with a $50 reusable reader and phone app for team sports. Data stewardship requires informed consent, on-device processing when possible, and transparent models to avoid biased clinical decisions. Like a library card system, data access is carefully tracked and protected while enabling valuable health insights. Accessibility considerations must include diverse skin tones, hair density, sweat rates, and socioeconomic factors to ensure equitable healthcare. Cybersecurity and tamper detection are essential for medical-grade deployments and insurance reimbursement programs. Post-market surveillance closes the loop from real-world field performance back to iterative design updates and improvements. Let's summarize the complete pipeline from laboratory validation to clinical deployment. The complete pipeline flows from laboratory calibration to clinical trials, manufacturing scale-up, and finally, deployment in clinical practice. Rigorous calibration and validation ensure measurement accuracy when converting between body fluids like sweat, blood, and saliva. Clinical use cases span the entire healthcare spectrum, from fitness optimization to chronic disease management and acute emergency care. Scalable manufacturing combined with strong ethical frameworks enables equitable access to this transformative health technology. The future vision is continuous, personalized health monitoring, accessible to everyone, enabling preventative care and early intervention.